developments in our own research, and how all this shapes or factors into the bigger Canadian landscape to really advance molecular pathology in precision oncology, primarily in pediatric precision oncology. So we all here are probably aware that precision medicine really started out as the idea of matching a certain drug to a particular genome variant. And so with this, moving away from a population-based approach or histopathology-based drug matching to a more molecularly targeted one. And this might be variants identified, on a larger scale, so that we know at a larger scale this variant works, or it might be in a more personalized context where we match this on a very individual patient basis. And so we essentially, if I get my pointer here, we essentially study at the DNR level, or now sometimes at the RNA level, and then find a molecularly targeted drug and want to change the cancer phenotype. And so the question is, how well have we done in the pediatric world in the last couple of years? These are data from 3 different clinical trials. Well, the first one is good. In most of the studied tumors, we find alterations. That's not surprising, given that these drive the malignancy. The good one is that in more than half of them, we actually find some that we classify as actionable. So where we think we have a suitable drug that might either attack that protein, encoded protein directly, or a related pathway or mechanism. So that's great. But if we then look at the number of kids that were treated, we are really dropping down to lower percentages. And then of those that were treated, only 10 to 50% actually had at least short-term beneficial response. So there is a huge gap, both in terms of the response, but also in the identification of actionable targets that we still would really like to improve on. And then, of course, a huge one that is related to drug and trial access to get more kids treated in the first place. And for us, really, the question is, what can we do to contribute to this? And if we look at what most molecular targeted cancer drugs really act on, it is the protein. And it's not the DNA or an RNA where we do our diagnostics on. And of course, we know that there are a multitude of processes that modulate the response from DNA to protein to the phenotype on its way. And so there's no straight correlation between DNA and protein levels. And in fact, if we look at this, so this is correlation data from a number of larger studies out of the US, but it's pretty commonly accepted now that the correlation of RNA expression levels to protein is modest as best, and sometimes even an anti-correlation. So there is no direct relationship between an RNA measurement and a protein amount, and there's even less of a correlation if we look at copy number changes and the protein amounts. So this allows us to hypothesize that we might do better if we actually look at the protein level directly and complement our molecular analysis, molecular genome and transcriptome sequencing, which now is pretty much established by other methods. And by pretty much established, I mean that they're relatively routinely done on children, at least of high risk or relapsed cancers. And we are now finally getting in a point where, as some of you might have heard, we actually get this paid from the healthcare system. So Quebec has led the charge here and introduced RNA sequencing coverage in recent months. And so we can hope that this now rolls across the country and we will see this happening. So the big question is, what is next? And we know that there are a number of very promising molecular analyses and assay systems on the horizon. Long read sequence, especially in the pediatric cancer world, can tell us a lot more about the structural changes, and a lot of the pediatric malignancies are driven by more structural than point mutation alterations. Methylation profiles or epigenetics in general is a big key that we have missed out on so far, and especially in some brain malignancies and sarcomas, carries a lot of potential. As we'll see quite a bit today, the proteome has a good potential to be the next complement to a whole genome and transcriptome analysis. And then, of course, we can get to a level where we've always been with immunohistochemistry. We can get back to a single cell and spatial understanding, so that we get to the heterogeneity of a malignancy. Or we move away from the tumor site itself and go to much more accessible test material and biopsy material, liquid biopsies, and what they can tell us about the malignancy at maybe non-reachable sites. So there's a lot that we would like to move closer to a routine application. Of course, there's a lot of work to done and quite a bit of red tape, and the question is, what can we do about this? So I'll talk to you about 3 stories today. First, about a first case of real-time proteome profiling informed patient care. Then we'll talk a bit about the concept of stability of the proteome and genome through cancer progression. And then lastly, about the recent developments in the Canadian Pediatric Cancer Consortium and where that might lead us over the next couple of years. So to start out, this story was really driven by Georgina Barnabas, a phenomenal postdoc in my group. And she really is leading the charge on everything precision oncology in the group, and has really made this story here possible. So if we really want to get good at a molecular analysis of a solid malignancy, then the type of specimen biopsy material is of course critical. And with, as you've seen, there are more and more assay platforms and assay possibilities coming up. And so the demand for specimens is getting bigger and bigger. And as I would think you agree with me, formalin fixed paraffin embedded material really has a huge advantage here over the need for fresh frozen. And that's for a number of reasons. One, we get a structural retention. We can look at the histology before we do a molecular analysis, but also we have the possibility, for example, in a relapsed malignancy, to go back to archived material from earlier relapses or the initial tumor of the same patient, because we do traditionally archive this material. And so this gives us a vast retrospective view into the history of an individual patient. And so what we are setting up here at BC Children's, together with John Bush, is a platform where we can scan and annotate the slides, and then do either macro dissection by scraping or micro dissection by laser capture. So we're getting a new laser capture in high throughput instrument into the Institute and into the hands of John Bush, and capture the isolated material and do sensitive liquid chromatography mass spectrometry on it for proteome analysis. And while FFPE is a fantastic material, it is not the easiest to extract protein from, but it can be extracted. The traditional methods were relatively time consuming and always required extensive sonication steps. We didn't really like this. It's not very reproducible, and it's not very automation friendly. And so Verena and Georgina really spearheaded the development of a really fast and streamlined assay, where the proteins are resolved by high acid concentration. The benefit of that is also that it gets rid of all nucleic acids. And then the D-cross-linking is removed. So the formalin cross-linking is removed by high heat and quencher. And then we move into a automated cleanup process and are ready for mass spectrometry analysis in a relatively short time frame. And that this works nicely is just here shown in a proof of concept across a number of different tumor types. They separate nicely on this PCA, and you see that we have in this particular analysis a depth of 5,000 to 6,000 proteins across a good dynamic range, and capturing what's highlighted here is a number of proteins that are either actionable or otherwise relevant in pediatric malignancies. And so you see proteases, you see epigenetic modulators, you see signaling molecules, you see receptors here. And so with that, we spend a good range of relevant pieces of information. And now when we do this, it's important to realize that what we do is quantitative, but it's relative quantification. So we don't go or don't reach absolute quantification and have no knowledge about the actual molar concentration of the proteins or number of proteins on a given cell. So we always need comparators, and one always have to think hard what a good comparator constitutes and how that affects the interpretation of the data, of course. So then coming to the question, is such a system, such a platform suitable to inform patient care in a clinical time frame? And we evaluated this over the last little while in a study, in a national study, the Precision Oncology for Young People that some of you might heard about. It's run and spearheaded by David Malkin at Sick Kids in Toronto. And it really brings together most of the pediatric sites and some of the young adult and adult centers, and addresses a number of questions, primarily genome sequencing for high-risk and relapse cancers. And out of UBC Pathology, we have Suzanne Carterin and Jonathan Bush leading the biobanking efforts in this area. We have Paul Sorensen, myself, driving the proteomics aspects together with Mike Moran out of Toronto. And so this is really a phenomenal platform to learn from each other and to move biopsy material around quickly, and to have molecular tumor boards in place in which we can learn from each other how different levels of molecular and modeling data can be interpreted and possibly inform patient care. And of course, we always prefer the established whole genome sequencing information that is already linked and proven, but in absence of this information, information of lower evidence levels can be considered. And that's what happened here. We had a young teenager who relapsed for a third time, I believe, and I won't go into the details here for privacy reasons, but that patient had a molecularly targeted treatment before. The tumor continued to progress again, and so was scheduled for an additional biopsy at BC Children's. The material was then directed to, of course, AP. We received H&E stained slides from them a couple of days later, and then within a number of 2 or 3 days, we could initiate the sample processing and mass spec analysis. And after overall 15 weeks past biopsy, we had an initial report and immunohistochemistry confirmation ready to be moved out. And then there was a bit of a waiting period to get the genome information assembled as well. And there was a molecular tumor board consultation about 2 months later. And then about another month later, innovative therapy was started for 2 months. And I'll talk a bit about this. So as I said, Formalin-fixed paraffin-embedded tissue has two benefits. One, we can go to archival material, so we could get information about the primary tumor and earlier metastases. And we also were able to separate out malignant areas from surrounding normal or adjacent normal tissue, which of course is not really normal, but gives you an ad hoc comparator. And so all the relative quantification data you'll see is compared to adjacent lung tissue. So overall, we got good quantitative information of about 7,000 proteins, and this is originating from a total of 82,000 separate measurements on these proteins. What we do is we split a protein into shorter peptides, shorter fragments of them, and then each and one of them will be measured. So for every protein, we have multiple individual evidences across the protein span, and with good dynamic range. So this is looking good. If we just look at this as a whole, across the primary tumor and the metastases, we see that the cell didn't dramatically change while it was metastasizing. So the primary tumor cells were still remarkably similar, vastly different than the normal material. But of course, so these are pathways, not proteins, but of course, some molecular mechanisms are changed after a cell underwent transition to metastasis and embedding in a new tissue. But you could clearly see that you're looking at the same malignant cell and cell of origin here. So the first thing we looked really at after we had that quantitative data and what Georgina did was to see if any of the treatments that we usually use in the context of profile and any of the molecular genome data we usually gather could be corroborated on a protein level. And so here you see a fold change of the malignant regions over the lung regions, and you see small log2 fold changes here. So none of the pathways associated to a genomic variant that we would usually use as a biomarker was substantially changed. P10 was actually increased while we would be looking for a P10 loss. So there was no corroborating data towards existing or usually used treatments, which was in a way disappointing, but allowed us to go deeper into the data and do more hypothesis-driven identification of possible new targets. And if we do that in an unbiased way, the one area that really strikes is that we have quite a bit of change in epigenetic regulation, which did put this in similarities to some synovial sarcomas here on an RNA level analysis. And then the other area that really stood out as really elevated proteins was the 1-carbon metabolism. And so Georgina looked into this more closely. And so here you see an excerpt of the 1-carbon metabolism pathway. And we have a number of key enzymes that are elevated here. So red is elevated log2 fold change over normal. And this serine hydroxymethyltransferase 1 and 2 really stood out. Here you see the absolute intensities compared. So clearly elevated in our samples, also across the historical archival material. And so if one dives a little bit into the literature and other preclinical data, this is really an emerging target in breast cancer and some other malignancies. And the challenge so far was that there was no good inhibitor that had a good bioavailability to be actually used in a patient context. But then a drug repurposing study identified sertraline, which is an antidepressant with safety data and approval for use in children and young adults as well. That was shown to be quite efficacious against SHMT2 as well. So that would be an alternative target for this drug. And it became apparent that most likely a single agent treatment will not have a sufficient response, and there were a number of other opportunities here, and that was brought forward to the molecular tumor board. It was discussed after, and sorry, I forgot that, after we went in and did a validation of SHMT2 by immunohistochemistry. This is not a clinical or LDT for SHMT2 because we couldn't find one. This is a piece of the work was really led by John Bush here. So we validated elevated staining in the malignant regions here. And we also looked across different other malignancies to see if, for example, we are in a range like for other malignancies where SHMT2 was proposed to be beneficial, like breast cancers here. And so we are clearly above that. So we had to resort to this immunohistochemistry across cancer comparison because we did not have a good proteome level comparison across cancers yet. And this is Ines in my group is currently working towards a proteome comparator database so that in the future we can go back to historical data on this and make it a bit faster. So the target itself seemed to be robustly elevated, and we confirmed there wasn't a fluke by the mass spec. Orthogonal validation is also nice, but we also wanted to have some functional validation that this pathway really was disrupted and that SHMT2 inhibition might help. And so this was in collaboration with Dr. Seth Parker at BC Children's, where we did stable isotope tracing, and he really confirmed a serine addiction of these cells in vitro settings. And we saw that sertraline in vitro did have a detrimental effect on these cells. And then there was some, in the context of profile, some modeling and expansion in mice, and also in the chicken aleontic membrane model. I'll not talk about those today. So based on this, the molecular tumor board discussed this, and it was decided to, a single agent sertraline therapy was eventually initiated. A multi-agent combination therapy was not feasible for a number of reasons, even though preclinical data would have suggested it to be beneficial. But as we all know, combination treatments are challenging to move forward. So where did that leave us? Well, these are CT scans of the patient, and you see here the regions growing over time. So this is before the treatment. This is while the patient was on single agent sertraline treatment. And if this is then after it was stopped and before a new targeted agent treatment selected by different means was initiated. And so here you see the growth rate at these different periods, and you see that there was a slight reduction of growth rate while on sertraline treatment, and it jumped back on when the treatment was stopped. But this treatment, this effect, while there, was insufficient to really benefit the patient in a major way, and it was stopped, and new opportunities were sought here. So this essentially leaves us with a problem here, and that is, well, one is the combination therapy that if there would have been more time to prepare for this, to get access to get additional preclinical validation that combinations would have been more powerful, could possibly have been a much more viable avenue. And that becomes challenging alongside the modeling to do really in this clinically meaningful time frame. And at the same time, it also shows the challenge that these families and the patients go through, that they have a diagnosis, they are treated, and then there's the phase of the monitoring. Maybe it's resolved, but oftentimes they relapse, and then it all starts over again. Essentially, we have to find a possible treatment, monitor, and possibly another relapse. And so it's this vicious circle, and also the fear of the next relapse. And so what's was proposed a couple years ago by the Better Response to Rhabdotomics Evidence study at BC Children's Hospital. So we initiated that study together with James Lim and Gregor Reed and Chris Maxwell to really see if we can prepare for a relapse. So if we can initiate diagnosis earlier, or molecular investigation earlier, do a thorough model systems analysis, and really have a possible treatment plan ready for an eventual relapse. This is economically questionable, but in terms of a pediatric setting, where the number of patients is luckily still relatively low, and the number of years lost or lived at reduced quality, and if one has to say it that way, also ability to contribute to society, is enormous. And so we haven't done the full economic analysis, but I think there's quite a good chance that this is actually economically viable if it makes biological sense. So the question becomes, how much does a malignancy change from diagnosis to a relapse, and do treatment opportunities we would identify early on still be present and relapse? And so that's a study that, so these are all the folks at BC Children's that work in this, again, from pathology here, John Bush and Suzanne Vercauteren. And so this is what Amanda Laurentian, a PhD student who just graduated and will be leaving us very soon to Genentech, in my group, really looked into, and now we are moving from solid malignancies to liquid, so to B-ALL and T-ALL in children, asking the question if treatment targets identified at diagnosis still matter at relapse. Because if that's the case, then we really can initiate this early. So she assembled a leukemia collection, a cohort of 71 samples at BC Children's, and also looked into genome data from 138 samples from St. Jude's Hospital. We looked at the level of whole genome and targeted sequencing of paired relapse diagnosis pairs. Later on some of these, we did paired drug response studies, and then proteome analysis was done on 50 of those, including 7 pairs, and so on. So at a high level, we see that the proteome groups the patients into a certain way, and what is becoming apparent, and the colors here are very colorful, but there's one cluster that stands out by having quite a number of events, following events. So this is either a death or a subsequent relapse. And so we were wondering how well classic risk group association classifies or predicts this follow-up event, relapse or death. And if we stratify by high risk and lower risk, so standard risk, then we don't see a significant difference in event-free follow-up over 5 years. But interestingly, if we grab this one or classify this, stratify this by proteome clusters, we see that this one cluster we also saw before really has a much lower rate of, a much stronger rate of follow-up events here. And now we're looking into this further. The honest answer is we don't really understand the biology of this, why this would be, what the mechanism behind this might be. It's a bit of a mystery to us. But there was a remarkable observation. But it's a bit of a side note anyway, because what we were really interested in is if treatment targets change or not. And so this is now looking at, so first focusing in, we have a couple thousand proteins, of course, quantified here, but we first were interested at about treatment targets and driving proteins, driving elements in those malignancies. So we focused on about a 250 protein set and looked if they were elevated in the diagnosis or the relapse or in both. And we find that the majority of these are elevated in both of the paired analyses, and only some show unique elevation. And here you see quantitative correlation of these proteins in the different patients. And that really, I mean, yes, there's a correlation and you get good numbers, but the correlation didn't make us really happy. It wasn't robust enough as an assessment to us. And so we were wondering how we could actually demonstrate stability of protein abundance more rigorously and statistically profound or relevant than in a mere correlation. And that's where Enes, bioinformatics, a graduate student in my lab, really had the brilliant idea to go to a statistical concept that's used a lot in psychology and in other areas, but rarely in molecular biology, and that's looking at equivalence. So we're all used to looking at difference, where we do a statistical t-test and ask the question if it's different from a non-change. But we rarely really investigate if things that do not change or are within a specific range of change are robustly in this change, or if our measurement precision is too low to actually make that claim. And so in addition to significantly different proteins, this equivalence test, which is a t-test against a different boundary, now allows us to say certain proteins are significantly within a certain boundary, so significantly robustly don't change more than a certain amount that you can define. And so if we do that, then and plot for every patient pair here the equivalence of proteins of all the ones where we can make a statistical assessment, we see that for our standards, wonderful, every protein is the same, is equivalent across our replication, and that's what we would hope for, of course. And if we look at a comparison of diagnostic cancers versus non-cancers, then non-cancer cells, then we see about 50% of the proteins are equivalent. And of course, there's some variation. Now, if we look at the same time points, so a diagnostic B-cell leukemia, for example, in different patients, we see that about 80% of the statistically quantifiable proteins are similar. But if we now look in the same patient at different time points, we see that we get up to 90% of the proteins remaining stable. So, of course, that still leaves about 10% of proteins that change and a few that we are not precise enough to make a claim. But point is, we see a remarkable amount of proteins, and that's now across the entire proteome or measured proteome of 5,000 or so proteins. We see a remarkable stability here. And if we look into the pathways that are the most stable ones, we see a lot of DNA repair, BC, and that fits to our story from before. We see histone acetylation, epigenetics, we see ribosome biogenesis and methylation. So some interesting areas here. Now, we were wondering if this is now actually a proteome effect, or if we would see the same at the RNA and genome level. because of course the prevailing concept is that we have clonal evolution that leads to a relapse, and that there are different genomic events that drive a relapse one. And we don't have data that would contradict it by no means, but it actually fits those models quite well. So first we looked at the RNA level, and now that's in a additional cohort, external cohort, and we didn't have enough information to do a equivalence test here. So we had to go by correlation, and what we can see is that we see strong correlation of RNA levels, and also what you see in black here is where a patient pair diagnostic relapse is the closest cluster neighbor. So you see there are quite a number of those pairs grouping together. And so it seems that this is not a proteome phenomenon, but that the transcript levels really add up quite well as well. And if we look at targetable mutations in those patients, we also see that a good majority of the mutations is shared. So it's present. If it's present at diagnosis, it will also be present at relapse. And some are only showing up at relapse, and some of this is clearly in response to treatment, like anti-5C2 here, for example. This only shows up after chemotherapy treatment, and this, of course, makes sense and has been shown before. And now, one of the reasons why we think a lot of these these targets actually don't change in a relapsed one is because we go in with a broad-scale chemotherapy, where resistance mechanisms are quite different than if we were to go in with a targeted agent. So, of course, if we were to go in with a targeted agent, hitting somewhere alongside the CDKN2A axis, then this would probably be eliminated at a relapse. So if we look at the agents we would choose based on this, you see agent or pathway target chosen at DX and relapse, and all the green ones are the ones where you would choose the same at diagnosis as you would at relapse. That covers 63% of the patients, and then some where you have nothing at diagnosis and only at relapse. So for a good number of patients, this actually holds true, and that's encouraging. Now, we were wondering if, okay, so the targets are there, you would choose the same targets, but is the drug response the same? So if we do a targeted treatment in vitro, based on agents chosen by the mutational pattern, do we see the same sensitivity in diagnosis as in relapse, and across a number of agents tested here against MEK inhibitor, CDK4/6 inhibitor, JAK-STAT inhibitor, and smooth inhibitor? We see that overall we have a good correlation here. But if we take a bit of a closer look at the presence and absence of the respective target biomarker in the patients, we, like for CDK4/6 inhibitors, for example, really the patient cells in vitro from those with and without the target do actually respond pretty similarly. So there's maybe this subgroup here, but that tells us, while the sensitivity is the same, really the specificity to this and the suitability of this agent matching is not particularly high. And that is probably part of the story to when I'm at the very beginning, I told you that we don't always see a good response to a targeted agent. And so in part, maybe we don't really do the best match here. And there are, of course, a good number of other reasons. So we were wondering if proteomics would actually suggest any additional drug targets we could go for. And so Amanda went in and was looking in her entire data for a protein that's present in most samples, a protein that's amongst the top 5 abundant proteins based on the rationale that if there's more of it, it's easier to really dampen far down. And then if it's the top 5 percentile of increased in cancer versus non-cancer. And so what really came out was part 1, which we identified in most. It was really in the top percentiles here. And there are PARP inhibitors available. And so we looked into 2 different PARP inhibitors, looking at non-cancer cells and leukemic cells in vitro again. And we did see a nice separation here. And we had started to initiate that in mice. That's a bit challenging. And so we're following up on this. So in the last few minutes, 5 minutes or so, I want to now talk a bit about what's happening at the national level, what we are building on this, and where we might move this forward. Because of course this is an N of 1, and this is a leukemia study. So the first thing, of course, is the stability that we looked at in leukemia. Now the big question for us is, does that hold true in a sarcoma? Does it hold true in a glioma? Or is this really specific to B-cell leukemia? Does it even hold true to, in an AML situation? And so we are expanding this to look at different malignancies here. So as I said, PROFILE is a national study, and there are a number of national studies out there that capture aspects of really the patient trajectory through treatment, but also of the research trajectory, really basic research, and then trying to translate it and trying to advance it towards clinical care. And that's where in about a year ago, patient advocates, people with lived experience, really went to Parliament Hill and managed to get an additional funding support out of the government, which then was handed over to CHR to create a Canadian Pediatric Cancer Consortium with an initial runtime for 2 years. Let's hope we can make this longer. And so the vision for, I believe not all of you have heard of that, so that's why I'm introducing it here quite briefly. And so the vision of this Canadian Pediatric Cancer Consortium is that every Canadian child with cancer will eventually have access to the latest scientific advances, diagnostic tools, therapies, and supportive care, leading to better outcomes and quality of life. And so what I highlighted here is what is most relevant to our department here, and this is latest scientific advances. So really, how can we make sure that the research findings get moved towards the children? And then how can we make sure that we have equal access to the latest diagnostic tools and not just standard of care testing? So the whole consortium is split up across 7 or by now even more groups. One that really looks at the basic biology questions, mechanistic questions, but also molecular pathology testing advances is cancer biology group, which is co-led by Nada Jabado and Sonia Saloa of Montreal and myself, and 3 phenomenal people with lived experience that really contribute a phenomenal piece to this work. And then relevant to what we do is also access to innovation therapy, a group where we would like to advance assay to more innovative assays. So out of our department, Suzanne, John, Paul, and myself are part of that. I'm hoping I'm not missing anybody. If so, let me know. And also, whoever wants to join this, this is really open and really open for everybody to contribute, to join in. That doesn't mean that everybody gets $1 million the moment you start, because the money is still limited. But it's a phenomenal group of people to really advance this forward and rethink some things here. So in the cancer biology, a few things are being done. First, we are initiating some national research studies in brain sarcoma and leukemia. Paul Sorensen, of course, heavily involved in the sarcoma space here and thankfully driving this. Really working with all the other national and local initiatives to improve our data gathering and biopsy sharing and access, so that we can, especially in a area of a rare disease with pediatric cancers still are, we really need to make the best out of the information we have and build bigger cohorts by sharing patient-derived models and phenotyping. Then it will drive real-time proteome analysis. I've shown you an initial case, and this will now be pushed into the next more or less routine analysis, research grade, but routine analysis at a similar level where RNA sequencing and whole proteome analysis have been for the last couple of years. Liquid biopsies are of particular interest. And then, as I said, and I'll spend some time on this, democratizing molecular test access. So to really ask the question, how can we get the next level, the next whole genome sequencing, the next RNA-seq closer to a clinical application, or where can we get tests that we have, lab-developed tests that we have in one lab, maybe at Sick Kids, maybe at BC Children's, make sure that that's accessible to everybody around the country and that everybody is aware of it. And then, of course, also for those where everybody is aware of the tests and know that they would be great, but where getting funding for this individual tests might be a challenge, might be too slow to get to, or even impossible to get to. And so how can we streamline that? Get research assays closer towards a translation and get lab developed tests to optimal use. And so it's still in early drawing stages, but a process that's being thought of is really that either the oncologist or the pathologist can identify a need for additional testing, because of course we won't need additional testing for every patient, but for some it will be highly relevant or promising. Then we have a standing molecular pathology board that can on a short notice evaluate those cases, and if a need and suitability is identified, then dig into a national registry for research assays and laboratory developed tests. So we're essentially every lab, research or clinical, can register their expertise and abilities and needs, what they would need for this analysis. And effectively what it becomes is a matchmaking. So we have a need and we have a possible test, and then we contribute the required funding for this. And the idea is doing this on a relatively short time frame and turnaround, so that then we can transfer the samples. And of course, we need to have appropriate study enrollment on a local or national study. But with this, we can possibly create a relatively quick pathway towards this. And then the important piece, of course, is to have biannual impact evaluation, where we can learn two things. One, which of research assays did really deliver valuable information and probably should be moved forward and maybe prioritized for development into proper tests. And then also, which of the tests that are not currently covered by the funder should maybe moved or suggested for a fee coding. Although that, of course, is again a lot of red tape and will take years, but getting solid data, solid impact data at a national level can really facilitate that. And with this, I would really like to thank all the people who not only did all the work, but also really made this a fun and exciting and engaging adventure. So really, all the people in my lab, you're the best. It's fun and it's exciting. In the two studies I talked about, Profile and Brave, and of course, John Bush and Suzanne Becotter, and yeah, everybody, really, the funders and patient advocates, people with lived experience. Without all those, it wouldn't be possible.
Thank you, Phil, for that nice presentation. Very, very exciting work and impactful. Anybody has any questions to Phil, please go ahead.
I can ask one. Torsten.
Yeah, Torsten. Hi.
Yeah. I hear this, and I want to work with you more. Be it on sarcomas or proteomics or test development, because it overlaps with a lot of stuff I do. But the question I had goes back towards the beginning of your talk with this sort of ominous graph of going to actionable mutations and then new treatments that you might try, and then response rates, and then everything dropping precipitously, and then what you didn't even try to show, which is survival, right? Which that actually matters, and again with the anecdotal case you showed. How do we deal with this issue when you have few patients, fewer who have this particular mutation, and accumulating the evidence to show that something really works and is worth paying for, and convincing the test and the treatment. And in the context, I know in the nucleic acid field, people can kind of pool data, right? They're using the same platforms. They can find ways to pull the data in the cloud, if you will, or registry. It seems that that's quite a bit harder with proteomics, isn't it? Because the techniques aren't standardized.
It is quite a bit harder. There's a lot of effort going towards standardization, so this is getting better. But it and there's also a difference between the whole proteome coverage and then if we know a particular target is really relevant and should be tested for a certain match and targeted agent, we absolutely can develop targeted assays for this, where we can do absolute quantification and they can be compared across labs. So this is possible, or one would switch it into an immunoassay at that point. So this can be developed at the point where we then have the data that, oh, it's always this protein that needs to be up, that we have a good understanding that this treatment would work. And Mary could, of course, speak much more and much better to this. But in terms of the data, I think you're right that we will have a few cases, and it will be disparate to say this treatment really works when this is elevated. But the more low-hanging fruit, the more realistic one is to gather information that on the patients on which a complex proteome analysis was done and where Molecular Tumor Board had molecular protein level data available, they did better or they did worse. And of course, initially it will be limited to event-free follow-up for a number of years, because of course survival oftentimes is a number of years later, unless we are in the really high risk ones where death would follow soon. But it is a challenge. It is a challenge, and it's early days and early steps. But we essentially faced the same challenges on the whole genome sequencing level. A bit easier on the standardization side, but on really showing evidence took a good number of years. That, I mean, only now are we getting in the point, at least in Canada, to make that. Suzanne?
Thank you, Philip. That was amazing. And it's kind of, I have a question on the same line as Torsten did. Like, for leukemias, we have such a good survival rate. How do you put more money into it for like few people? Like, how do you identify the people who really need that extra workup with genome, proteome, the whole meal deal, versus patients who totally don't need it? Like, the vast majority of our patients do really well.
It's a very good question, and it's in many ways it's a societal question. I guess the one question is, how well do the patients really do after a heavy chemotherapy? or transplant. Yes, they absolutely do survive, and they live reasonably well over a long time, but it's not without a cost. So one can see it as a research investment into possibly eventually replacing some of the chemotherapies. And this can only be done if we also start looking early for the ones where we have the treatment right now. But really for leukemias, an important piece, I think, is also this being fast. And the getting proteome level information is possible in this 2-week or 3-week time frame that we have, even for more rapidly progressing leukemias at a relapse stage. So that's at a point where we already know that they won't do well. They are the small fraction that don't do well. And so that's absolutely a point where we can start to gain information at moderate investment. And maybe we learn that this is so beneficial that it really justifies doing it earlier as well. Maybe not. Maybe it's in that space, it's really limited to those, but do it fast.
Yeah. And as a parent, like every investment is worth your child, of course. I'm not saying that we shouldn't be doing this. I'm just saying kind of a lot of the kids do well. And how do you say to taxpayers, we're going to increase your taxes for a 95% survival rate? But sure. Just being controversial, sorry.
No, absolutely. I mean, those are the discussions we have all the time in as you know.
Well, thank you, Phil, for the excellent talk. I think I have to go to another meeting.
There's somebody in the chat. Oh, okay.
Yeah. Anybody has any questions can talk to Phil privately. Okay, thank you all for attending this.
Thank you, Dr. Gao, and thank you everybody for coming out.
Enjoy a long weekend, everybody. Thank you.
You too. And yes, let's talk, Torsten.
Yeah.
Absolutely. Take care. Bye-bye.